Hi, my name is Sarah Gravem. I am a research associate at Oregon State University in the Partnership for Interdisciplinary Studies of Coastal Oceans, PISCO. It's a mouthful, and I study marine intertidal ecology. So I study how organisms interact with each other and interact with their environment. And one of my main parts of my research is studying how climate change is affecting all the little critters that live in the tide pools. Well, on a fieldwork day, I get up at about 3 and I eat a big breakfast and I drive out to the coast, get here by dawn, and spend 5-6 hours out hitting that low tide to get as much fieldwork in as I can before the tide chases me back out and I can't access my research subjects anymore. So here we are in the Rocky Intertidal. This is the Intertidal Zone along this transect line that I've laid out. And so right here next to the water we have the low zone where you see tons of different types of algae. There's some sea stars, sponges, sea anemones, sea urchins, just tons of different organisms all living together. And as we go up the zone, you see these guys, these big clam-like things, mussels, taking over. They are the bullies of the intertidal. They grow fast, they settle in the millions, and they're big and tough, and they kind of push everybody else out. As they take over, they change the whole makeup of the community that's below our feet. And as we get higher and higher and higher, you can see that the mussels disappear toward the top. And that's because it gets super hot in the daytime. So what I'm studying here is how climate change is affecting all the animals that live in this intertidal zone. And so what you can see is that those mussels, for example, have this low zone boundary here, and above us they have a high zone boundary. And so what we call that is a zone, and the mussels are pretty much in the mid zone of the intertidal. And so at the bottom, they are cut off, and at the top they are cut off, and that is their entire ecological niche. You can walk from one end of their ecological boundary, their range boundary, if you will, to the other end in a matter of steps, which is super duper cool if you're studying something like climate change and how it affects species ranges. So this is a sea star or a starfish. It's called an ochre star. This is our common sea star on the west coast. They come in orange, purple, red, brown, and if you ever have been to a west coast tide pool, you probably have run into them. These are actually voracious predators that eat all sorts of stuff. They're the top predator, and they're so important that we actually call them the Keystone Predator here. And the reason for that is because their favorite food is mussels, and you can see this guy is actually eating a mussel, or just ate a mussel. It is spitted out, it's all digested, and that's just the shell left. And so these guys kind of live in the lower zone. So we just covered how at the top of the zone there are all these stressors pushing mussels down, and at the bottom of the zone there are predators pushing the mussels up. And what I'm interested in is how this ecosystem is going to look in 10 years, in 50 years, in 100 years, and how climate change might change all the zones that we see here. So, for example, if we're talking about mussels, warming climate could do a couple things, right? First, it can push down the upper limits, right? So you've got warmer air, more heat waves, more stress, pushing them down so they'll die at the top and survive, say, in the middle. We also have sea level rise, which is going to make the whole intertidal zone go up, which isn't my expertise, but certainly something we think about. And then at the bottom of the zone, you might think, oh, climate change shouldn't be a big deal down there, right? Because that's the less stressful part. That's the part they're in the cushy ocean. But the lower limit of the mussel bed is the same, essentially, as the upper limit of their predator. And so whatever climate change is going to do to this predator will affect the lower limit of the mussel bed. So the way that we measure how communities are changing over time is pretty actually low tech, at least on its surface. So this is a transect line. I have bolts that I drilled into the rock, and they're from the low to the high, so I can place this tape at the exact same location every year, and I've been doing this for 5 years now. And along this tape, we can use quadrats, and we use photographs, and we use all sorts of other things too, but we can just lay this down in the same spot exactly every year, take a photograph, and then analyze how many sea urchins, what's the cover of algae, how many mussels, how many sea anemones, and we can use this to look at how the beds are moving down, or biodiversity is changing, or the numbers of sea stars is increasing or decreasing over time. We also look at how the environment changes, and so this over here inside this little cage is a temperature logger. So this is actually recording temperature all year, every 15 seconds. And that's important because the temperature in the intertidal changes really drastically. You go from being underwater and being at like 8 degrees Celsius, 10 degrees Celsius, to on a hot day, this can get up to like 20 if on a really, really hot day. And these organisms that live here have to deal with this extreme fluctuation in temperature, like at least seasonally and often daily. But there's one other big way that climate change is affecting these systems, and that's through disease. And so sea stars, these guys, actually in 2014 here in Oregon, went through what was a sea star wasting disease, Epizootic, which is epidemic for animals, zoo. This is the largest marine disease on record, we think. The wasting disease started in 2013 in California and Washington, and over the next couple years spread all the way from Mexico to Alaska. The death rate in Oregon for these guys was between 60 to 90%. So at this site, I think it was around 90% of all the animals that lived here died. We were super interested as scientists to understand how this loss of a keystone predator, the top predator in the ecosystem, which was like continental scale within a year or two of each other, affected the community. And I mean, it's pretty rare that as scientists we get to watch how an ecosystem changes due to the loss of a top predator, when we have a before picture, a during a picture, and an after picture. And one thing that we're really focused on is how ecosystems are resistant or resilient to that change. And so we're able to gain insights into how all ecosystems work. We have an ability to use the things that we find here and infer what's happening in a national park in the middle of America or a coral reef in Indonesia.